Hi, Mrs. Ortega here. I am getting ready to take a look here at Chapter 42, where we will cover circulation and gas exchange. So every organism must exchange materials with the environment. As we've seen through these vertebrate and vertebrate chapters, every animal we've looked at had some way of exchanging materials with their environment, getting food, getting oxygen, getting carbon dioxide for plants. I mean, every organism, living organism, needs to exchange materials. And so in previous chapters, we looked at unicellular protists and how they exchange directly with the environment. And then we moved on to multicellular animals where direct exchange is not possible. And so there is a need for a specific feature or organ to perform this function. And so in this chapter, we'll look at that internal transport and gas exchange that's very functionally related in most animals. And so we'll start out with the circulatory system and how circulatory systems will link the exchange surface with cells throughout the whole body. And so looking at circulatory systems, you want to remember how exactly does the oxygen and the carbon dioxide, how does the oxygen we breathe in and the carbon dioxide we exhale get to every single cell in the body? And the answer is the circulatory system. Through the tiny network of capillaries, these red blood cells containing oxygen will transport that oxygen to every body cell. And so in this chapter, we'll look at how that occurs. But it occurs differently in different animals. So we'll take a brief look at other animals as well and how they have different systems. So gills are an example of an exchange system that is going to be in an aqueous or aquatic environment, whereas lungs will be an air environment, atmosphere environment. And so looking at the circulatory system, we want to first define what type of circulatory system it is. There are actually two main versions of circulatory systems found in animals, and these have different adaptations with each animal or species, but overall there are two main types of circulatory systems. And those are open circulatory systems and closed circulatory systems. But both of these have a circulatory fluid, a set of interconnecting vessels of some sort, and a muscular pump or a heart. And so the circulatory system connects the fluid that surrounds the cells with the organs that exchange gas, absorb nutrients, and dispose of waste. And so the circulatory system has to connect with not only the respiratory system, but the digestive system and the osmoregulation excretory system. And so it's a very extensive system. It needs to have many vessels or or a different way with lots of connections to these body circuits. And so looking at open and closed circulatory systems, the circulatory system that's open is going to be found in some insects, arthropods, and mollusks. And this open circulatory system is going to have a fluid called the hemolymph. Since it's not the normal consistency of blood, it doesn't have the same components, exactly the same components as blood, it's not going to be called blood. And so it's called hemolymph. And so in this, we'll see an example here where hemolymph bathes the organs and then circles back to the heart. Whereas in a closed circulatory system, blood's going to be confined only to the vessels, and it's going to be distinct from the interstitial fluid that's between cells and between different organs and different tissues. And so there are annelids, cephalopods, and vertebrates all have closed circulatory systems. So here we see the example of an open circulatory system in a grasshopper. In the overall diagram of the grasshopper here, you can see that there are pores. These pores are connected, moving from the anterior to the posterior region on the dorsal side. And these pores are the tiny circles that are on the tubular heart, which are the larger cylindrical shaped valve shaped things there. They're connected to valves. And so what happens here is that the heart will pump the hemolymph out of these vessels. These vessels will spread that hemolymph over the organs and bathe those tissues in this hemolymph. And then relaxing the heart is going to open up those pores that are on the heart for the hemolymph to circle back into the heart. And so those pores on the heart have valves. And so when the heart contracts, those valves are closed. So it's able to push out the hemolymph to the body. And so body movements will also help circulate the hemolymph, as it relies on body movements, as it does not have anything else to assist in circulating it back around the organs and back to the heart. Compare that to your closed circulatory system that you see here, where there is a heart that pumps blood through a vessel. It goes from the vessel to the capillaries and then back to a larger vessel that returns blood to the heart. And so this is very distinct region. These vessels are in very distinct regions, separate from the interstitial fluid, the watery matrix that's in between cells. Our example here is the earthworm, but there are lots of vertebrates and other animals that have a closed circulatory system. And so we will look at the organization of vertebrate circulatory systems next. Humans and other vertebrates have the closed circulatory system that's called the cardiovascular system, where there are three main types of blood vessels. Those are the arteries, veins, and capillaries. And through all of these blood vessels, blood flow is only one direction. So that we will see why that's essential, that blood flow will be only one way in these vessels. And so taking a look here in a little bit more detail, the arteries are going to be the largest blood vessels that carry blood away from the heart. These will branch into smaller vessels called arterioles. And then eventually these will branch into capillaries. So arteries are the largest, then arterioles, then capillaries. And so arteries and arterioles carry blood away. So I like to think A equals away. It's carrying blood away from the heart. And blood vessel flow is based on the heart because that's the easiest way to define it. Looking at capillaries, you will see networks of these capillary vessels called capillary beds. These are going to be a site of chemical exchange. So this is where the oxygen is going to enter the body cell, and then the body cell is going to send carbon dioxide back to the blood in the capillary bed. And so that's the chemical exchange. Oxygen going into the body cell, leaving the blood, and then carbon dioxide entering the blood leaving the body cell. Venules is the next one here. Venules are the large vessels that will return blood to the heart. These are venules are the smaller, scratch that. Venules are the smaller blood vessels that will then converge into veins, which are the larger vessels that return blood to the heart. And so arteries carry blood away, veins return blood to the heart. And so this is organized by direction of blood flow, not by oxygen content. And so we will see how vertebrate hearts have 2 or more chambers. And in the heart, there will be an atrium that will allow blood to enter and be temporarily stored. And then it's going to be pumped through a ventricle chamber, which is a muscular chamber. And so in vertebrates, we'll look at how the circulatory system operates. The first one we'll see is a single circulatory pathway. This is done in bony fish, rays, sharks. And so they will have a 2-chambered heart with a single circulation. And so in this, blood will leave the heart. It will pass through 2 capillary beds before returning. So let's take a look here. And single circulation, first the blood will fill up in the atrium, and then the atrium will open its valve to the ventricle, where then the blood gets pumped, pushed out of the ventricle into the artery that's carrying blood away. This oxygen-poor blood will then go to the gills in this fish, where then it exchanges carbon dioxide and oxygen. And now oxygen heavy, it's going to then go to the body capillaries, where it spreads out, exchanges that oxygen with carbon dioxide, and then converges back into veins, where then it goes back to the atrium. So single circulation, 1 heart pump, goes through the gills, then goes to the body, then goes back to the heart. Now let's take a look at double circulation. In terms of double circulation, amphibians, reptiles, and mammals all have the circulation pathway. And so in this pathway, oxygen-poor and oxygen-rich blood are going to be pumped separately from the right and the left sides of the heart. And in reptiles and mammals, oxygen-poor blood will go to the lungs or the pulmonary circuit. In amphibians, it will actually go through a pulmocutaneous circuit through the lungs and the skin. And so we'll see that here in just a moment in picture form. But in terms of delivering that oxygen to the body, it's going to be done through the systemic circuit. So delivering that oxygen-rich blood to the body is done by the systemic circuit. So this double circulation allows for a higher blood pressure to be maintained than single circulation. As that single circulation, the blood pressure drops significantly going through the capillaries. Anytime blood has to go through the capillaries, the blood pressure will drop. And so it drops significantly getting to the gills, and then again going through the body capillaries. So looking at double circulation of an amphibian with a pulmocutaneous circuit. Here, pulmocutaneous circuit. First we always start, I believe, at the sorry, I feel like I'm dyslexic right now. At the right atrium, which is here. When looking at the heart, they are defined by the viewpoint of the organism or the patient. And so it's always backwards to what you think it would be. So this that I just circled is going to be the right atrium, which for us is on the left side. And that does not make sense, but is how it's labeled. So we will continue that way. And so starting with the right atrium, oxygen-poor blood gets stored up temporarily, then gets pushed through the one ventricle that's here, the one ventricle, and from there, for the double circulation, the amphibians, they actually do have a ridge in this purpley region that prevents or keeps about 90% of the blood going to the right direction. And so that's going to keep most of the blood going to the lungs through the artery here is going away. And then it's going to circulate through the lungs or the skin. And so as frogs can breathe through their skin, that is done, especially when they're underwater. They will redirect all of their oxygen poor blood to the skin, where then it will exchange the gas. And then from there, it's going to go into the left atrium, which will temporarily store that up, and the oxygen-rich blood store it up, and that's going to go into the ventricle and pump out of the ventricle into the artery that's going to go out to the body. And so here you can see in the systemic capillaries is going to go to the body, exchange that gas, and then go back to the right atrium. And so for mammals, it does look a little bit different, as we have a 4-chambered heart, not a 3-chambered heart. For mammals here, you can see a clear left and right atrium and left and right ventricle, and the direction of flow is the same, except it has a pulmonary circuit. So pulmonary, pulmo means lung. It translates to lung. So that is how you can identify the circuit. It's going to the lungs, whereas systemic is going to the body. For the previous one here, pulmocutaneous, pulmo meaning skin, pulmo meaning lung, and cutaneous meaning skin, like subcutaneous, if you've ever heard that. Injecting something subcutaneously means into the skin. And so that means lung or skin circuit. Alrighty. So now we're going to take a look at how the double circulation and heart contraction drives the cycle in mammals. And so this is going to meet the body's need for oxygen at all times. And we'll see how the mammals and the birds have the 4-chambered heart, 2 atria and 2 ventricles, and the left side of the heart is going to pump oxygen-rich blood, whereas the right side of the heart is going to receive oxygen-poor blood. Right, and so here this page describes in word form what we're going to see in the next image. And so if you have questions, look at this page. I'm not going to read that to you. I'm going to describe it as we look here. So starting out with your right ventricle, this is going to have the oxygen-poor blood. This is going to be pumped. This muscle is going to contract and squeeze at that bottom here, and then pump it out through the pulmonary arteries. So if you're wondering, you do not have to necessarily memorize the name to know where this is going. So if you know the arteries go away, then you know that this is an artery, both of these here. And where is it going? To the lungs. So that's pulmonary. So these are pulmonary arteries. In the pulmonary arteries will lead to capillaries in the lungs, where gas exchange occurs. And then that oxygen-rich blood is going to want to return back to the heart from the lungs. So returning to the heart will be a vein, and coming from the lungs will be pulmonary. So these are pulmonary veins. And from there, it's going to go to the left atrium, where it's going to temporarily fill up there in blood. That's going to send it to the left ventricle, which will then have the largest muscle here to pump that blood out of the heart through the largest blood vessel, which is the aorta. And then from the aorta, it's going to go to the coronary arteries that will then lead to the rest of the body. And so these arteries will take the blood eventually to capillaries in the upper and lower body regions that will exchange the gas of carbon dioxide and oxygen, and then the blood will return. And so returning blood from the upper half of the body will be through the superior vena cava. And then returning blood from the lower part of the body is going to be from the inferior vena cava. That will then go to your right atrium. And then from there it goes back to the right ventricle. And so now we're going to take a closer look at the mammalian heart. So there are two atria and two ventricles. The atria are going to have very thin walls, and these are serving as temporary collection chambers for blood. And so this is for blood returning to the heart, and it's about to be pushed back out or pumped out through the heart. The ventricles are going to have much thicker walls and contract much more forcefully than the atria, as that they are responsible for pushing that blood out to the body or to the lungs. And so this happens in a cycle of contracting and relaxing, and that's called the cardiac cycle. And the contraction or pumping phase is called systole. It comes from a Greek word that means to contract. And so that is going to be the contraction phase. The relaxation or filling stage is called diastole. And so diastole means to pull apart or to relax. And so that's where it's also Greek, and that's where we get that stage name. And so here we can see the mammalian heart, a closer look at the anatomy of the heart. The right atrium, starting out there, you can see it has a what we call it atrioventricular valve or AV valve that goes to the right ventricle. That's a valve that has three parts of it that help open and close that valve. And so that's also called a tricuspid valve. And so moving out from the ventricle, the right ventricle, to the pulmonary arteries, there's a second valve called the semilunar valve that's there. And this prevents backflow of blood, that would keep blood in the heart otherwise. Looking at the left atrium and the left ventricle is going to be the thickest muscle of the heart, as it's responsible for pumping blood to the whole body. And it has both the atrioventricular valve, which here is going to be bicuspid. And then the semilunar valve as well, that's pushing blood into the aorta. And so how does that cycle happen, you may ask? And that's what we'll see here. The cardiac cycle starts off in atrial and ventricular diastole. So this is a complete relaxation phase where the ventricles and the atria will fill up with blood. And then about 0.1 seconds later, the atria are going to enter contraction phase, the systole, and then the ventricles will be still in relaxation phase. And so that atria closing out is essentially pushing as much blood as it can into the ventricle, and it's shutting that valve closed. And so when it shuts that valve closed, it actually does so forcefully that it makes a sound. And so that sound is the first half of the sound you may recognize from a heartbeat, the lub sound. And then from there, about 0.3 seconds later, the ventricular chambers will be in systole, so they will contract. They will push out that blood through the semilunar valves and into the artery and into the aorta. And so this contraction phase is going to also have a closing of those chambers, the semilunar valves. When they close off so forcefully, it's going to have a dub sound. At this stage, the atria are already going to be in diastole, and so a relaxation phase filling up. And so as soon as that is done, about 0.4 seconds later, it's going to now be filling up all both atria and the ventricular valves, and so or chambers. And so that's the cardiac cycle. So the first part is filling up the heart, then the atria contract, making the lub, then the ventricles contract, making the dub, and then it goes back to filling up again. And so as you can imagine, there's a lot of study, a lot of definitions and qualifiers about the heart and the circulatory system. And so here we see some of those. The heart rate is also called pulse. It's the number of beats per minute. And that's an average of 72 beats per minute for an average person at rest. The stroke volume is the amount of blood pumped in a single contraction, which averages about 70 milliliters of blood. The cardiac output is the volume of blood pumped into the systemic circulation per minute, and that depends on the previous two measurements. And it's an average of about 5 liters per minute. So every minute, all of your blood gets cycled through once. And so that's a very large output. And so we have those 4 valves we mentioned that prevent backflow of blood into the heart. They're described here. The atrioventricular valves, or AV valves for short, separate the atrium and the ventricle. The semilunar valves are going to control blood flow into the blood vessels from the ventricles. So blood flow into the aorta and the pulmonary artery from the right and left ventricle. So we discussed the lub-dub sound already, and so that's there. And then any backflow of blood through a defective valve may cause what we call heart murmur. So it's actually pretty frequent to be born with a heart murmur or a defect in one of the valves that causes some residual blood to remain behind. It can be operated on, but it can also be something that still allows for people to live full, healthy lives. So it really depends, I guess, on the defect and how significantly it impacts the heart. So now that we've looked at the heart and structure, we want to identify some features about the heart beat. So it has an autorhythmic beat, meaning it can contract without any signal from the nervous system. So as we look at this here, you will know from experience your heartbeat can change based on your situation. And so that is done by the nervous system. But in its normal resting state, it does not need any help. And so that will be autorhythmic. And so looking at the heart rhythmic beat, we'll start out with different regions of the heart, one of which is called the sinoatrial node, or SA node for short. It is also commonly called the pacemaker, as this area sets the rate and the timing at which cardiac muscles contract. And so impulses from this node travels to the second node called the atrioventricular node. And if you were to guess based on the name, that would be somewhere around the atrium or atrial and the ventricular valves. You'll be correct. And so at the AV node, then this impulse gets delayed for a little bit, then travels to a region that we call the Purkinje fibers that make the ventricles contract. And so then the ventricles contract, and so impulses that travel through the heart, making it beat, can be recorded in what we call electrocardiogram, ECG or EKG for short. And so here you can see the control of heart rhythm in diagram form. First, starting out, the signals in yellow from the SA node, which is the tiny region here. Oh, can't circle correctly. And the left atrium in that back area, kind of close to where the superior vena cava will connect. That is the SA node, the pacemaker region. That will start the signal. It will send it to the AV node, which is by the atrioventricular valve of that area. And then it's going to have a slight delay where it's going to send that signal through the bundle branches into the heart apex, so the bottom of the heart here, and then it's going to then spread throughout all ventricles using the Purkinje fibers. So reading these signals on the ECG below, you can see there's definite region of communication or signal from SA node spreading through the atria. That is called or abbreviated as the P interval. Then there's a Q interval, that is when the signals are delayed at the AV node. The R interval is going to be when the signal is spread throughout the ventricles. Q. I missed Q. Q is there in between with the bundle branches. And then S and T are follow-up after contracting of the ventricles, then the relaxing of ventricles, and I believe the S is the delay of signal. And so I've labeled those on there for you here on the ECG. These are commonly known for medical professionals, and anywhere from doctors to pharmacists to scientists use this information to monitor, track changes of the heart. From personal experience in a pharmacy-based region, a pharmacist can look up and see how the medications might affect regions, of which I have a personal example of where we saw a patient whose R to T interval was being delayed so much that it was actually going to, he was at risk of early death because of all the medication that, when combined, prolongs this interval too long. And so we obviously took care of the situation, but this is widely used in many different applications and many different settings. And so it's very important to understand just the process that occurs, not necessarily how to read a ECG or EKG. But I find that pretty interesting and hope you do too. So in this section, in the rhythmic beat, the pacemaker is regulated by the nervous system. So the sympathetic and parasympathetic divisions of the nervous system, we'll see this in chapter 49, these divisions, but they both do regulate the pacemaker. One of them, the sympathetic, speeds it up, and the other one slows it down. But the pacemaker can be regulated by hormones and temperature, providing a higher speed or lower speed when needed. And so now we're going to take a look at patterns of blood pressure and blood flow and how this reflects the structure and arrangement of blood vessels. So the blood vessel's cavity is called the central lumen. So this is where blood is going to be flowing through. It is called the central lumen. The epithelial layer of tissue that lines blood vessels is called the endothelium. This is smooth. It minimizes resistance. It's usually made up of simple squamous cells. If you remember from chapter 40, I believe, we looked at the cell types. Simple squamous cells. And so here I put together a table of the structure and the function of the capillaries, arteries, and veins. And so we'll see this in a picture form in the next slide. But this is a very great way to study in terms of understanding the differences in the structure and the function of these blood vessels. And so starting with the capillaries, they have the thinnest walls. They are, the diameter of the capillaries is going to be slightly wider than the width of a red blood cell. And so they can only pass one red blood cell at a time through that part of the blood vessel or the capillary. In terms of structure, they have the endothelium and a basal lamina surface. And so it's very thin and it's functioning exchanging the materials. So the nutrients, the gas, carbon dioxide, oxygen, hormones, other things like viruses, bacteria can function exchange through the capillaries. For the arteries, we have there are thicker walls in the arteries. They have an endothelium, which is spelled incorrectly. I'm sorry. Do take a look at the correct spelling there. It has thick, smooth muscle. As arteries are carrying blood away, it's going to need to keep that very high blood pressure. And so to do that, they're going to have a thick, smooth muscle that's going to help propel that blood along. And then they have a layer of connective tissue that's going to connect it to other organs and other tissues in that area. Then veins are going to have thinner walls and a thinner, smooth muscle, as they will have other methods of moving the blood back to the heart. We'll see that their valves are uniquely designed, and so they don't need as much smooth muscle on their blood vessel structure. And so you can see those in picture form here. Arteries, the endothelium lining, the thick, smooth muscle, thicker artery wall. Then capillaries are just the endothelium and basal lamina, the connective tissue to hold it in place. Veins, thinner connective tissue, thinner, smooth muscle, and then the endothelium layer. I believe, let's see. We do have another image with the structure of the vein in terms of returning blood to the heart we'll get to. So blood flow velocity is very important to look at blood flow and blood pressure. And so we have a diagram to depict that here. In terms of area, centimeters square, the area is going to drastically increase in the capillaries. So the amount of area that the blood flow is going to cover significantly increases. And so in the arteries and the veins, that area is going to be much, much lower as they're centralizing the flow back to the heart. velocity is going to be a very high starting out, but when it reaches those capillaries, it's going to die down. So thinking like traffic, the velocity going through the arteries is going to be very high, so very little traffic on those roads. And then when it hits the capillaries, it's going to be hitting rush hour traffic, and it's going to be waiting, one blood cell after another going through those capillaries. And then the veins returning it back will slowly but surely get better. So a slow increase in speed in traffic would be what you would see with velocity. Looking at pressure there, so pressure is going to be measured in terms of millimeters of mercury, and that has the two pressure points of the heartbeat, which is systolic and diastolic pressure. So systolic is when it's contracting, so it's going to be higher in number than the diastolic, and so it's going to alternate, and that's going to be very high throughout the aorta, the arteries, the arterioles, and then it's going to slow down the capillaries, venules, and veins. And so as it gets farther away from the heart, as it gets very, very low, and as when it returns to the heart, after it returns, it's going to resume at a high blood pressure. And so looking at blood pressure, it's going to exert in all directions, including against the walls of blood vessels. And so the resistance to blood flow in the narrow diameters of tiny capillaries and arterioles will dissipate much of that pressure. And so looking at systolic pressure, that's when it's the ventricles are going to be in contraction phase. That's the highest pressure in the arteries, whereas diastolic pressure is going to be when it is when the arteries, or when it's going to be in diastole, when it's all going to be, the heart's going to be in the relaxing stage. So it's going to be much lower. And so you can feel a pulse is a rhythmic bulging of those artery walls within each heartbeat. And so blood pressure can be regulated through our own homeostatic mechanisms. They also can be regulated through other methods, artificial means. But here you can see vasoconstriction is going to contract the smooth muscle, increasing blood pressure. It's going to make those blood vessels smaller. Vasodilation is going to be relaxing smooth muscle, so making those blood vessels bigger, and it's going to cause blood pressure to fall. And so there are some natural vasodilators and vasoconstrictors that you can see here, but these aren't anything to memorize. What we will want to focus on is the blood pressure and gravity. So blood pressure and measuring blood pressure is usually done in the arm at the same height as the heart. The wrist blood pressures are also widely available. However, they're not quite as accurate as holding it, positioning it correctly is going to provide either really a good reading or an off reading. So there are other ways to measure it, but looking at measuring the arm is going to be best. And so looking at the measurements themselves, the blood pressure for a healthy 20-year-old human at rest is 120/70. The common average seen worldwide is 120/80. That's taking in all ages into account. And so for us, we'll look at in terms of 120/70. And so how do you measure blood pressure, you might ask? With one of our measuring devices that you see in this diagram here, using a cuff, an inflator, and also a stethoscope, you can measure it by inflating the cuff with air. This will establish a higher pressure around the arm, and this is going to actually temporarily cut off that artery in the arm when the pressure is equal to or greater than the pressure in the blood vessel. And so closing that artery, it will be greater than 120. Then you slowly decrease the air in the cuff, and so this will drop the blood pressure, will drop the pressure in the cuff. And so looking at the blood pressure, you're going to want to listen for audible sounds in the stethoscope. When blood starts flowing through, it's going to make audible sounds, and so those sounds will let you know what the systolic pressure is, which is 120. So systolic pressure is 120. And then when you keep decreasing the cuff until the sounds stop, and that will be the diastolic pressure. And so for humans around 20 years of age, it would be 70. For others, it could be 80. And so keeping a track of your blood pressure is good to maintain your health. And so it is important for us to understand what these measurements are and how to do so. So systolic is a contraction phase. It's higher in pressure. It's 120 on average. Diastolic is a relaxation phase. It's lower, and it's going to be average about 70 for this age group. And so looking at the veins, returning blood flow back to the heart from the lower half of the body can be a challenge, as it's going against the force of gravity. And so to do so, it's going to have not only valves along the vein, and it's going to use skeletal muscle to help push that blood back towards the heart. So overall, blood is moved through veins by smooth muscle contraction, skeletal muscle contraction. Also, the expansion of the vena cava with inhalation will pull blood back. And so you can see the valves here will open and close for blood flow, and that will prevent backflow of blood. And looking at capillaries, capillaries are going to function for gas exchange. And so here you can see there are two mechanisms for regulating blood distribution in capillary beds. While this is important, it's not something I ask you to memorize. And so you can see here there are pre-capillary sphincters that can open or close, and that will regulate blood flow through the capillaries. And so here you can see as blood flow, you can see the direction, the arterial end of the capillary and the venous end of the capillary. Blood pressure goes down, and osmotic pressure goes into the capillary. What we will see here for capillaries is how it correlates with the lymphatic system. So the lymphatic system will return fluid that leaks out from the capillary beds. So it's fluid, that blood plasma, along with blood cells, can, especially with rips and tears in the capillaries, can leak out of the capillary. And so the lymphatic system will be responsible for returning it back to the circulatory system. And so here you can see the lymphatic system drains into veins into the neck, where they also have valves that prevent backflow of fluid. And so any swelling caused by disruptions of the lymph flow is called edema. Lymph nodes are going to play a role in filtering this and in defense. And so here you can see how a diagram of how that lymphatic system will collect and then return the blood and other blood materials back to the circulatory system. Let's see here. I'm going to go ahead and break this video up into two just to make it easier for y'all. So I'm going to stop here and then start again very shortly.